of the Coalition Against Childhood Cancer. I welcome you today along with our guests who are presenting a really exciting and emerging presentation about a new research study that was published about G-CSF and cancer stem cells, and I really appreciate you giving your time today. I will let you know that we begin with all of our listeners on mute so that any ambient noises won't be bothersome to our panelists. There is an opportunity on your control panel to the right to submit written questions at any point during the presentation. We are recording the presentation, and once we have approval from the various panelists to be able to post it, we will. And otherwise, I would like to ask the slide presenter to move on to the next slide so that I can remind you, like we do in every meeting. Jason, can you move that to the next slide? Thanks. The CAC2 values. We start our presentations this way to remind everybody that in this business, what we're trying to do is put children and their families first in everything we do. That's why one of the reasons why we were eager to sponsor this presentation. We also try to be inclusive and collaborative, assume positive intent, and all these other values that are on the slide. And so that's just, we have a lot of guests today who are not CAC2 members. We welcome you, but we also welcome the CAC2 members. Just a little housekeeping, besides the idea that you're going to submit your questions in writing, we also want to remind you, if you haven't yet registered for the annual meeting that will take place next month on June 16th and 17th in Washington, D.C., you should be wanting to do that because space is limited. I am joined today by Scott Kennedy, who is one of the parent co-founders of Solving Kids Cancer. He has also had the privilege of being the father to Hazen, who was a neuroblastoma patient and fought cancer until the age of 5. Scott's background is in the pharmaceutical industry. I also want to acknowledge Trish Kreiger of the Jeff Gordon Children's Foundation, who is our driver and technologist today. She does a splendid job and will be the one accepting your questions and moving them on to the askers. So we appreciate all her work. And at this point, I will turn the conversation over to Scott so he can introduce our presenter and welcome you all as well. Scott?
Thank you, Vicki. I want to thank you and Trish and all of CAC2 for your partnership in hosting today's webinar. I think it's an example of the values that you just posted about collaboration. So we're really grateful to have you as a partner be a member of CAC2. I also want to thank everyone that's on this webinar for joining in so quickly. We feel a sense of urgency in opening up discussions like this that have such significance in the day-to-day care of our children. And so the hope here is that for all parents, including myself, to wrap our heads around the trade-offs and the decision-making that comes with this great responsibility of being the ultimate caregivers of our children. And so to help us, we're proud to have Dr. Jason Choate joining us today as our presenter. I just want to give a brief bio. Dr. Choate is an MD, PhD at Texas Children's Hospital and an associate professor in the Department of Pediatrics at Baylor College of Medicine. Dr. Choate is also the research co-director of the Neuroblastoma program at Texas Children's. And his research focus includes developing better molecularly targeted therapies for neuroblastoma as well as other solid tumors, as well as the translational efforts to test specific inhibitors and novel platforms like nanoparticle drug delivery. Along the way, Dr. Choate has developed a particular research niche characterizing novel stem cell population in neuroblastoma, as well as creating new hypotheses on how neuroblastoma cancer stem cells may have a role in the progression and relapse. I think his ultimate goal would be to develop new therapeutic approaches that address this. Dr. Showitz published several research articles, including the recent study on G-CSF, cancer stem cells, and a potential role in neuroblastoma progression and relapse that will be the basis of today's webinar discussion. And before I turn it over to Dr. Showitz, I want to thank him for his time and his cooperation in leading this important discussion so quickly after the high-level summary of his findings spread rapidly throughout the childhood cancer community. So thank you personally and from all parents and all the charity leaders, Dr. Showitz. So without further ado, I want to present Dr. Showitz, Dr. Jason Showitz, to discuss G-CSF, cancer stem cells, and relapsed neuroblastoma. Dr. Showitz?
Thank you very much, Scott. I really appreciate the kind introduction, and I'm very excited that there's so much interest in our research. I'd like to mostly use this time to clarify some of our findings and also put them in context of the current clinical practice for neuroblastoma, which involves using G-CSF as in supportive care to support neutropenia in our patients. So I'm going to jump right into a little outline of what I'd like to talk today about, and first I'm going to describe, just so we have an idea of what I'm talking about when I talk about cancer stem cells and how we found them in neuroblastoma. I'd also like to briefly go over the data that's published, that we just published and that we previously published about cancer stem cells. And specifically, I'd like to definitely emphasize what we've demonstrated in mouse models and in cell culture and what we have not demonstrated. And also follow that up with the purpose of G-CSF, describe a little bit about its functions in normal cells and possibly in cancer stem cells, and conclude. And really, our data is just the start of what we've uncovered some new biological information about neuroblastoma, and I want to make it clear what we're recommending and where we're going to go forward from here at Texas Children's, and I think in other labs and clinics across the country. So I wanted to start with the fact, or multiple studies have shown in multiple different types of tumors, that despite what cancers look like underneath the microscope, as shown here, this is an MIBG scan and a CT scan of a child with neuroblastoma. And under the microscope, the cells all look very homogeneous. They look like small blue cells with some matrix in between. But we've learned over the past several years that tumors are they're not all the same cells. And that if you dig into the differences between individual cells in a tumor, you can see that they have different surface receptors. They're growing at different rates. They're communicating with each other at in different ways. And so a primary, in this case, a neuroblastoma, or many other cancers are described this way, as groups of multiple different types of cells, as shown here, different colors. Some of them are stroma derived from the host. Some of them are more differentiated. Some of them are less like nerve cells and more like primitive precursors. And then there's a subpopulation. These red cells would be considered a cancer stem cell population. And a stem cell population has a very specific definition. And cancer stem cells have very specific characteristics. So a stem cell is what we can think of as a progenitor cell. They're a cell from which the entire tumor can evolve. And so this cell, a stem cell, can make multiple other types of cells, as well as self-renew and maintain its own population. So stem cells generate other types of tumors, all the subtypes within the tumor. They generate tumors from very few cells, at least that's the hypothesis. And they self-renew themselves. They maintain themselves despite what's going on with the rest of the cancer. They keep themselves going. So that's what a cancer stem cell is. That's what a normal stem cell is in our body. We have stem cells in our blood. We have stem cells in other tissues. And so the study that we just published is really a follow-up study of a paper that we published in 2013, which was mostly the effort of a very persistent and I guess persistent is the best way to describe. Danielle is a pediatric surgical resident at the time. She's subsequently gone on to be a surgical attending, who really pursued the earlier observations that maybe the G-CSF, maybe neuroblastoma expresses G-CSF, and it might, we pursued the hypothesis that maybe the G-CSF receptor could be detected in neuroblastoma. So this earlier paper showed that we can in fact purify a subpopulation that expressed the G-CSF receptor out of neuroblastoma tumors. We showed with some very meticulously that this subpopulation is much more tumorigenic than the rest of the cells that we purified, and in terms of forming tumors in mice. And I left a bullet point out. G-CSF receptors, positive cells, were always present in all the tumor cells we looked at. And in general, they made up a small percentage of the tumor, usually less than 1%, although that varies from tissue to tissue. And just to emphasize, we found them in all the cell lines we looked at. We looked at multiple different types of xenografts of tumors growing in mice, and importantly, from primary patient biopsy specimens. Once we confirmed that every single cell line had a stem cell population or had these G-CSF receptor positive cells, we looked in primary patient biopsy specimens with the help of our surgical colleagues at Texas Children's. And we also showed, one other point we showed was that, at least in the mice, when we treat the mice with chemotherapy, it seemed like we could enrich for these cancer stem cell populations. And this is just a table from our earlier paper that showed that all of these are patients before they've received any treatment whatsoever, and they all have somewhere, this is 0.8%, this is 0.6%. They all have a small percentage of these G-CSF receptor positive cells. We did have some samples that were taken after treatment. Usually we resect tumors after 3 or 4 cycles of chemotherapy. Those tumors tend to have an enriched population, 3%. This one had 9% G-CSF receptor positive cells. So basically, this earlier paper showed that there's definitely a subpopulation in neuroblastoma that expresses the G-CSF receptor expresses the G-CSF receptor, excuse me. The rest of this paper was taken up by looking at the stem cell phenotype of these cells, which I'm not going to go into today, but at the time, a few years ago, you really had to prove that something was a stem cell, and there were very strict, very clear definitions that had to be met in terms of, for us to call them cancer stem cells. So what did that paper not show? It did not show, we didn't really show function of G-CSF binding to this receptor. And we didn't show that, does that alter the behavior of the cells, or does it alter their ability to form tumors, or does it cause the tumors to spread or not spread? We didn't have any of this functional characterization of what happens when the G-CSF receptor G-CSF, excuse me, binds the G-CSF receptor. I just want to point out that and this will come up later, and this is one of the, I don't have a lot of pathway slides or anything, but this is just a diagram of the G-CSF receptor on the surface of a cell here. Once G-CSF binds to the receptor, it activates a very potent downstream signaling molecule called STAT3. And STAT3 is involved in stem cell proliferation and stem cell self-renewal. Both normal cells and multiple different types of tumors have shown that cancer stem cells signal with STAT3. So this signaling pathway made a lot of sense for a cancer stem cell model. So it took us about a year and a half to get more data to prove some of these things, and that's what we most recently published. It's available. This paper is online in Cancer Research, and what we did in the most recent work is we used several different animal models. Again, both human tumors growing in mice, and we also used mouse tumors growing in mice. And we found that if you give G-CSF to these mice that have tumors, that the tumors were bigger, grew larger. There was more metastasis to the bone marrow, and we've also shown this to the liver now, and we have more metastatic data that we have not published. The tumors were bigger, and importantly, the larger the tumor, the more cancer stem cells they had. We also found that giving G-CSF increased the amount of cancer stem cells that were in the tumor. So that is very interesting. We also have some data in the paper showing that if you target these stem cells, or if you block G-CSF with an antibody that prevents it from binding to the cancer cells, that these tumors grew much smaller and they didn't spread. The other thing, as I mentioned, the importance of STAT3, there are small molecule inhibitors, chemicals that you can inhibit STAT3 with, and those turned out to be very effective treatment in our mouse models for the entire tumor. I think so. This is one of the only data slides I have, and I know it's a kind of busy slide, but this is just sort of our one experiment, a description of an experiment. I thought it would be good to show you with the kind of experiments we did. This is a mouse being injected with neuroblastoma cells into their renal capsule, and then we gave either VP16 or etoposide plus this inhibitor STAT3, plus or minus the inhibitor, and we gave multiple cycles of chemotherapy, and then we looked at the size of the tumor and how much tumor had spread to the bone marrow. And this cell line, this was the NGP cell line. It's an NMYC amplified cell line, pretty typical of a lot of the different neuroblastoma tumors. If you inhibit STAT3 signaling, the tumors grew very small. They grew a little bit, but they were very small. If you gave chemotherapy, there was a range of responses. If you combined chemotherapy with the STAT3 inhibitor, the tumors were much, much smaller, and this is highly statistically significant. We also showed that when you give chemotherapy, there's a slight increase in the cancer stem cell population, but most importantly, when you gave the STAT3 inhibition or the combination, we could not detect any metastasis in these mice in their bone marrow. And this was using a quantitative PCR assay for the NMYC gene, so it was very, very sensitive assay for metastasis. So really, we showed that blocking G-CSF signaling plus chemotherapy was better than chemotherapy alone, at least in these mouse models. And we did this in several different cell lines, several different types of mice. So for the purpose of this conversation, I really want to emphasize what we did not show, okay? Because I know people are concerned about, well, is G-CSF hurting our kids? Is there a problem? And we did not show in this study that if you give G-CSF, that if you combine G-CSF with chemotherapy, that this alters chemotherapy, or if it makes tumors grow more or less resistant to chemotherapy. We haven't done those studies, and that's sort of an ongoing project. And so right now, we have no evidence that G-CSF changes the effectiveness of chemotherapy, at least in our animal models, okay? We just want to emphasize that we haven't done those studies yet. There's certainly ongoing studies that need to be done. I think it's really important that we sort of expand our efforts to do some of these studies and look at what happens when you do multiple cycles of chemotherapy with and without G-CSF, sort of as we do in children. What happens? Is there an effect on metastasis rates? Do the tumors, if we make the tumors go away, do they come back faster or slower? then does G-CSF alter drug resistance in the neuroblastoma that does come back? And there are multiple other in vivo testing that needs to be done to just determine what does G-CSF, how does G-CSF interact with chemotherapy? And also in a longer discussion that we probably don't have time to get to today, I'll touch on a little bit, is what's the best way to target the cancer stem cells? Because if we can target the cancer stem cells specifically, maybe we can prevent some of the metastasis that we see and some of the cancer recurrence after chemotherapy. Why do we use G-CSF? I think it's a really important question that is very clear, and all of the clinicians across the country know why we give G-CSF, and the main reason is that we need to support children who have very low levels of white cells. G-CSF is going to cause their white cells to come back faster. And outlined here is just a diagram of the lineage that G-CSF works on. As I mentioned, there are blood stem cells. G-CSF causes granulocytes, which are the cells that fight infection, to grow faster and come back faster after chemotherapy. So one of the main points is that our current chemotherapy is dose intensive. So dose intensity is a concept where we give large doses of chemotherapy close together, and the timing of the cycles between chemotherapy may be important in terms of curing cancers in general. Dose intensity is very important because it should kill more cells with each cycle of chemotherapy and should prevent cells from growing back between cycles. The other thing is that dose intensity exposes children to prolonged periods where their white cells are very low, and it's important. G-CSF is very important to reduce the amount of time that patients are exposed, or that patients have very low white counts, to prevent life-threatening infections. So G-CSF is very important for how we give our current chemotherapy, and we don't have data saying that when we give G-CSF, we're making our chemotherapy not work as well. I just want to emphasize that. So where do we go from here? What are our main conclusions? First, as always, we don't want to do any harm. We don't want to cause children to suffer life-threatening infections. And I think some of the points that we're emphasizing is that our current chemotherapy, okay, has achieved the highest cure rates ever. And many of these treatments include G-CSF for their safety reasons I just mentioned, and also to permit dose intensity. So that's our current chemotherapy, and we're doing really well. We could do much better. Could we improve this? Of course, but we're doing well with G-CSF. That said, our studies in the laboratory have uncovered new biological information regarding this cytokine, this growth factor, and its influence on neuroblastoma cells in animal models. We're not going to stop using G-CSF, but it's very important that our lab and other labs study the effect of G-CSF in neuroblastoma. And I just want to emphasize that G-CSF is permitting our current best therapy. So, if I put on my PhD hat, I want to say we've discovered, as we discover new things about cancer, we need to try to incorporate them into therapies as, excuse me, as rationally as possible. From an MD hat, I put on my MD hat, I say, well, we have to make sure our children are safe, and we don't want to cause any harm by just reacting to studies we've done in the lab until we really are sure what the best way to proceed is. And at Texas Children's, currently we don't recommend people stop using G-CSF. All right. As I said, we're going to continue to study this, we don't want to compromise our current treatment intensity by delaying chemotherapy. I think it needs to be studied. If we use less G-CSF, there are many things to look at, but we don't want to put children at risk for life-threatening infections. And I think with that, I'll conclude. I have, there are other things I can show you, but I think it's most important that we answer some of the questions that the audience has, and some of the questions. I'm very happy to answer any questions that I can. So thank you.
Thank you, Dr. Showit. This is Scott again. It's a really comprehensive discussion. I'm sure it's prompting a lot of questions. We got some submitted already. I want to remind all the participants that we set aside this time to address as many questions as possible. It looks like we have about 20 minutes for that. So if you do have questions, please submit them now or throughout the discussion that we're about to have. We really want to express as many of your questions and concerns and comments as possible. So we encourage you to submit the questions through the questions section on your panel on the right side. Dr. Showit, I'm going to start it out with a basic question. You talked about the animal models and that this work is based on things done in mice. So what is your opinion about how reflective the mouse models would be of the same as in the system of children? And the second question that's related to that is the dose level that you gave in those experiments equivalent to what would be given with current G-CSF administration?
Okay, those are two really, really good questions. So the first part, if I could rephrase it, how well do our mouse models reflect the human disease? And I think, so there are several, it's actually a very long answer, but a short answer is that our animal models are pretty good at some things, and they're not as good at other things. So the tumor microenvironment is very important. So by that I mean the other cells that are growing around the tumor in a child or in a mouse are very important for how the tumor cells interact and how they grow. When we look at cells in tissue culture, they really don't grow the same way as tumor cells in a body. And mice they may not seem like close to a human body, but they have similar stroma cells, they have similar immune cells, and they have a similar level of oxygen, for instance, and temperature, etc. That being said, the mouse models don't necessarily reflect the immunological status of the human, and especially our xenograft models. These are immunodeficient mice, so they don't have T cells and B cells, and so that also is an important factor to take into account. So mouse models are only as good as the questions you ask of them, and I think we're always trying to make better tumor models and animal models for neuroblastoma. And the second question was about the dose of G-CSF that we used. So that's a very good question. It's something we struggled with for a while in order to set up these studies. So there's a couple basic things. First of all, mice metabolize G-CSF about 20 times faster than humans. So they crank through it. And previous researchers have looked at G-CSF, have given G-CSF to mice to look at other diseases, autoimmune diseases, and to study infectious diseases. And we used doses that are basically equivalent to what previous people have used in mice. In data that we didn't publish in the paper, the G-CSF level of dose that we gave to these mice increases their white cells modestly, but it didn't cause massive proliferation of their white cells. and they tolerated it pretty well. So we can only estimate that it's, that it's somewhat similar to humans, but really there's no way to directly compare it.
Okay. Another question related to also using a similar thing in standard neuroblastoma treatment is G-CSF, which is used along with anti-GD2 antibody therapy. The question is there a difference between G-CSF and G-CSF? And then secondly, are you finding the same findings with G-CSF as well?
Okay, actually I have a slide. There is your answer to your question. So G-CSF is completely different than G-CSF. So this is the G-CSF receptor. It looks totally different. It's got a different structure than G-CSF. G-CSF also stimulates white cells and also monocytes. So it's granulocyte monocyte stimulating factor. G-CSF sort of binds to a different receptor that we have not found in neuroblastoma. We haven't exhaustively looked for G-CSF, but we couldn't detect it by flow cytometry. And so really G-CSF is not an issue from our standpoint in terms of cytokines activating neuroblastoma. That's the short answer.
Okay. G-CSF is also used for other pediatric cancer types in for the same neutropenia improvements. And so is the relevance you found with the neuroblastoma cancer stem cells also applicable in other pediatric cancer types?
That's a question I can't answer. Every different, like, osteosarcoma, Ewing sarcoma, the other solid tumors, we haven't looked exhaustively for this cytokine receptor in those tumors. The cancer stem cells for different types of tumors are still being defined, and I think that what researchers are finding is that different tumors have completely different stem cells. So I don't think, I can't comment if G-CSF does anything to any other solid tumors.
Okay. And then I have a question that covers a lot of concerns and comments and questions from several families that perhaps aren't even participating in the webinar, but in a general type of way to cover a lot of things is, what should oncologists be advising families who bring up concerns about the use of G-CSF?
I would just reemphasize that I want to reiterate that we're recommending that we continue to do what we're doing. We have a lot more work to do in the lab and possibly in the clinic to try to figure out ways to address, determine the real clinical significance. I think, like I said, our best cure rates are with our current therapies, which are dose intensive, and they pretty much require G-CSF. And so we're recommending, no change at this time. Clinical practice evolves according to what we know, and what we discovered that ALK is something we can target. When we give ALK inhibitors to patients with ALK mutations, this is another thing that we may be able to target, and targeting this subpopulation is something that I'm working really hard on, and I think we'll get there in the future, but right now we just have to, I think clinical practice has evolved. It's based on all of the things we've learned to date, and we have a safe, effective way of giving chemotherapy, and I'm not certainly recommending any changes to that.
Okay. And I have a couple specific questions, and maybe we'll have some other more general questions too, but this is more of a specific one. Is it more significant to avoid use if that's what the thinking is of where to choose to use G-CSF, in particular after stem cell transplant for any particular reason?
You mean the use of G-CSF after? Oh, okay. So after stem cell transplant to help engraftment.
Right.
No, we don't have any data suggesting that is any different than our use for after chemotherapy. I mean, there are. I'm not a transplanter. There are ways to help engraftment that may not use G-CSF. I don't know that, but I think, yeah, we don't have any data saying that that context will be different than what we've already shown. Does that answer your question? I'm not sure. I have an answer to that.
I think we could, if there's a follow-up to that where someone feels it's not answered, they can use the follow-up email, which is a poll and also provides an opportunity to ask questions, and the email address of info@solvingkidscancer.org for any follow-up, if that's necessary on that question or any other. This is another specific question. Are there any trials that are currently open that use STAT3 inhibitors going after the G-CSF receptor? So STAT3 inhibitors, that's a very good question. There are STAT3 inhibitors in pharmacologic development, and there have been some clinical trials of a few different STAT3 inhibitors. As far as I know, to date, the data of those drugs has not been very exciting, or they have, all of the drugs so far have had different problems. But that's something we're really interested in is STAT3 inhibitors. It's definitely what we think. It's definitely a new therapeutic target for neuroblastoma, and we're working on that intensely. So okay.
Yeah, I don't have the latest on STAT3 inhibitors. There may be clinical trials I'm not aware of, but they haven't been ready for prime time yet.
Another very specific question is this question around the use of G-CSF. Does it relate in any way to MIBG therapy for relapse?
I don't think so. I think MIBG can be very effective in different settings, and actually the stem cells should be as sensitive to the MIBG as the non-stem cells. So yeah, I don't think there's going to be a connection there. But that's certainly something we want to look at and try to model in the future.
Okay. Is there any thinking? There's a question here. I'm just summing it up, but it's asking about how G-CSF. You're saying that it doesn't reduce the effectiveness of chemotherapy if chemotherapy is fighting more cancer stem cells and large tumors. So how can that be? Let me see if I can understand this one. Let me come back to that one because I actually have to reword that. Okay. Is there any data? Is there any data that's related to specifically any clinical data about the use of G-CSF and relapse rates?
No, there is no data. So one of the problems with our current data set, our clinical data set, is that G-CSF was introduced in the mid-1990s, and it kind of crept into clinical practice. So there isn't a cohort of patients that with comparable chemotherapy and intensity that we can compare that got G-CSF or didn't get G-CSF. There was a European trial that looked at G-CSF. That looked at their question was, can we give intensive chemotherapy, and if we do that with G-CSF, does G-CSF decrease the toxicity of the therapy while we're giving it? And so they that's the Siopin study by Ruth Lendenstein, and in that study they showed that if you routinely give G-CSF after every cycle, and you compare that group of patients to patients where the G-CSF was given under there were some rules about when to give G-CSF, and it was sort of up to the clinician to give it. That the patients who all got G-CSF after every cycle had less, they had some less acute toxicities. So they had less GI toxicity, less mucositis. There wasn't any, interestingly in that study, there wasn't any difference in infection rates or patients dropping off of the trial between the two sample populations. And then there's really no follow-up data from that study in terms of differences in outcome. I think actually there wasn't any difference in outcome between the two populations, but that really wasn't the question they were asking. Both, they weren't asking does G-CSF change the relapse rate. They were just looking at acute toxicities of the induction chemotherapy. And so, and it was a short-term study. They didn't follow those patients very long. So we can't really, personally, I don't think we can conclude anything about the effect of G-CSF on those patients' outcomes from that study.
Okay.
So there hasn't been any other study. I mean, it just, no one has thought about that study until actually we've, our findings really.
Going to a more general question, what should a parent or parents think about when they're considering that G-CSF allows their child to have more intensive chemotherapy and more frequent recovery so they can get the next round? And then this idea that's in the back of their mind about the same stimulant potentially causing progression. What kind of things should they be thinking about or talking to their doctor about?
Well, I'm sorry. Can you repeat that? Like, what's the?
Well, I guess another way to look at it is there going to be any type of materials or resource available that you think may come in the near future that would provide guidance so that these kind of questions and concerns that parents would have would be something that's easily discussable or available for them to talk to with their own doctor?
I think it's like any other medicine that we give to our patients. I mean, when we give Cytoxan, we talk about the risks and the benefits of Cytoxan, and I think you have to, physicians need to talk about the risks and benefits of G-CSF. It's pretty clear that right now, with the data we have, that the benefits far outweigh the risks, and I think that's all I can say right there. That's what we do as clinicians. We go over the risks and benefits of every medicine.
Dr. Showett, this is Vicki Binger, and I just wanted to let everybody know that we are approaching the time that Dr. Showett has committed to be with us. I think that we can convince him to answer another couple of questions before we wrap up. Scott, if you have some queued up for him, I think we have time for two more. And so just to let everybody know that we will wrap up, but as Scott said, you can expect two things following this call. One is that fairly quickly you'll receive an email with a short poll about your impressions of this webinar, but also with an open response question that if you have follow-on questions or comments that you can continue to have the conversation. And you can also, if you think of something later or miss getting the poll, you can send emails to info@solvingkidscancer.org. So Scott, go ahead and help us out with the next question or two, and then we'll conclude our presentation.
Thanks, Vicki. This is a question, I'm sure, from a concerned parent. It's they're saying that a lot of parents have used G-CSF to increase CD34 for harvesting stem cells. He or she wants to know, is this recommended? And then also, could cancer stem cells also end up being harvested too?
That's a very, very good question. And that's something I'm personally kind of worried about. But we need, it's something to look at. We don't have any data about that. The harvesting, giving G-CSF to harvest stem cells is very important. And we certainly recommend changing that. But I don't know. There's some work to be done there in terms of the best way to harvest stem cells for transplant. So the answer is I don't know, but that's a really, really interesting and important question.
Okay, great. And for the sake of time, I'll just ask one more question and reiterate what Vicki said, is that we have the questions that everybody submitted, and we'll do our part to share them with Dr. Showett and provide some feedback post-webinar so that everybody feels that their questions are listened to and answered. One of the last questions that came in, which is a great one, is there any alternative to G-CSF that would also help with neutropenia but not cloud the questioning of any connection to progression?
Okay, so alternatives to G-CSF. So GM-CSF, as I mentioned, does increase your neutrophil count. GM-CSF is not nearly as good a drug because it has side effects. It causes pulmonary edema. It's got some other side effects, toxicities that G-CSF doesn't have. There's no depot form of it, so that's something that children would have to get a shot every day of for GM-CSF. So yeah, I don't know of a good alternative to G-CSF for neutrophil support.
Okay, thanks, Dr. Showett.
Sure.
Well, I think that this was time well spent today. Thank you all for both CAC2 members and members of the general community for attending our webinar. If you have follow-up questions, or if your question, if we didn't have time for it and you would like to submit it again, please do so. On behalf of the Coalition Against Childhood Cancer, I'd like to thank Dr. Showett and Scott Kennedy and Trish Krieger for being our driver. There was a lot of questions, and I'm sure that in the coming days and weeks, more answers will be forthcoming. When and if this presentation is approved to be made available, we will post it to the cac2.org website. And look forward to seeing all of our CAC2 members in Washington, D.C. in about a month. Thanks very much.
And I'd like to say one more thing. I'd like to thank Solving Kids Cancer and CAC2 for putting this together. I really appreciate the opportunity to get some of this information out there. So thank you.